Recovering DNA from bones and teeth is a delicate and lengthy process that involves dedicated lab spaces, trained personnel, and specialized techniques designed for ancient DNA research. We begin with a bone or a tooth sample from an ancient individual. These samples come with explicit permissions from local communities, the collection institutions such as the museums that house these, and the government that provides permits for such research. The sample itself will undergo 3 important steps to go from bone to DNA sequence. The first step is subsampling, which involves the careful removal of a small portion of the sample for analysis. Subsampling occurs in a clean room, which for ancient DNA research is a specially engineered, well-controlled laboratory space with special filters to limit contamination by outside modern DNA. The sample is placed in a hood and carefully cleaned. The researcher then uses a fine-tuned drill to carefully remove the top surface of the bone sample in powder form. This powder is what goes on for further analysis. The original sample is then returned to storage and ultimately the museum or community of origin. The researcher then takes the powdered sample, only about 50 micrograms of material, into another space within the clean room to perform the second step in this process, DNA extraction. This procedure involves digesting the bone material, breaking open cells, removing proteins and lipids, and finally purifying DNA. Once the DNA has been isolated, it is then quantified to determine if there is enough to move forward, and if it's pure enough that downstream experiments will not be affected. The third step in this process is library preparation or construction. This step takes the DNA and modifies it so that it will be compatible with the sequencing system. For next-generation sequencing technologies, the sample generally undergoes fragmentation of the DNA, followed by attachment of oligonucleotide adapters to the ends of the DNA fragments, and finally quantification of the library product. Fragmentation is necessary for short-read sequencing, which, as the name implies, only reads fragments of a short length, such as 100 base pairs. However, ancient DNA is already fragmented by damage over time, so this fragmentation step is not needed. Oligonucleotide adapters are unique, known short sequences of DNA that allow DNA fragments to adhere to a sequencing instrument. Unique sequences called indexes or barcodes are also attached to the DNA fragments with the adapters to serve as unique identifiers of each of the samples being analyzed. Once the library construction is complete, samples are sent for sequencing at university core laboratories or companies that specialize in this service. DNA sequencing technologies come in many forms, but all of them involve determining the order of nucleotides in DNA. Once sequenced, researchers use the data for their analyses, whether that's to address questions about human population history, community structure and relationships, or pathogen evolution. Once completed, researchers will share their findings with the local community and the larger scientific community as a whole. For example, here, Mario Apata is explaining the research plan for a project studying the population history of the Camarões Valley in northern Chile.